Hi, this is Evan Blake with the World Socialist website. Today I'll be interviewing Dr. Amy Proal, who is the President and Chief Scientific Officer of the Polybio Research Foundation, the Director of the Long COVID Research Consortium, and a member of the editorial group of the John Snow Project. Dr. Proal is a microbiologist with extensive experience researching infection-associated chronic disease, including myalgic encephalomyelitis, chronic fatigue syndrome, or ME/CFS. During the pandemic, Dr. Proal has become one of the world's leading experts on long COVID, also known as post-acute sequelae of COVID-19, or PASC. Long COVID is a group of health problems persisting after an initial COVID-19 infection, which can impact nearly every organ in the body, can last for weeks, months, or even years, and are often debilitating. Dr. Proal, thank you for joining us today.
Thank you for having me on.
My first question is, can you share a bit about your background and what led you to get involved with studying viral persistence and long COVID?
Sure. So my interest in studying persistent infection with viruses, but bacteria and even fungi, a lot of different kinds of pathogens, actually stems from my own life, where when I was a student, a college student, I actually got a serious Epstein-Barr virus infection and didn't recover after that infection and developed chronic symptoms. And when I tried to understand why I developed chronic symptoms, there was a void. There was very little research on the chronic consequences of infection. And I began to even think to my extended childhood when I was trying to understand what had happened to me. And when I was a kid, between the ages of 3 and 5, I grew up outside the country, and I'm not sure what happened because I did get vaccinated for MMR, but I got measles and was hospitalized for that on one occasion. I got rubella and was hospitalized for that. I got scarlet fever and was hospitalized for that. I went through this slew of very serious childhood infections. I had a fraternal twin sister that did not get them, and I wondered if maybe some of those pathogens that I had gotten as a child had actually just not cleared from my body and were part of what were causing problems later. And so I began to read a lot of textbooks and just ask around and talk to researchers in the space who were studying persistent infection, which at the time and still is a smaller community of researchers than it should be. But gradually from there, I got a PhD. I did some experimental treatments actually aimed at targeting viruses and some cases bacterial pathogens in my own case, and was able to get a microbiology PhD and pursue that, and now connect research teams to conceptualize projects that continue to study how different pathogens and their failure to clear from the human body can contribute to chronic symptoms.
That's really fascinating. And what year was it that you got your PhD?
In 2011.
Okay. I guess I was wondering, I had a question if you could speak a bit more on sort of the history of the science of viral persistence. I know that there's a section of scientists, including yourself, who have known about this for some time. There was the landmark 1986 study, Persistence of RNA Viruses in the Central Nervous System, the pioneering work of Diane Griffin and many other scientists, but as you said, it sort of hasn't become mainstream even today, and many professionals, medical professionals and scientists are still not aware of these issues or well-versed on the science of long COVID. So could you maybe comment a bit on this history and sort of how the pandemic has impacted our understanding of chronic infections?
Definitely. So some viruses that humans acquire over a lifetime or commonly can acquire are studied fairly often in a persistent capacity. So for example, those are the DNA viruses, the herpes viruses being prime examples of those pathogens. So a lot of people are familiar with Epstein-Barr virus, or literally herpes simplex 1 that can be acquired, passed from person to person, depending, from close contact. Those viruses, which have a DNA backbone, have been studied as persistent pathogens, and it's understood via a decent amount of research studies that even when they persist, they can go through periods where they're dormant and they're not causing much problems, and they can remain in a basically silent state, or they can, under conditions of stress or immune depletion, they can activate and they can produce protein and drive a range. they're increasingly connected to a range of chronic conditions. But the RNA viruses, SARS-CoV-2 is a single-stranded RNA virus. Those viruses have been understudied in that same capacity. So in other words, there's a decent amount of an assumption that when people get an RNA virus, single-stranded RNA virus infection, that it goes away after they get through the acute phase, and that does not stick around in the body the way that a herpes virus would. But as you mentioned, if you look through the literature, there are teams that have studied the persistence of RNA viruses. And you mentioned Diane Griffin, who's at Johns Hopkins. She's a member of the Long COVID Research Consortium that we're now pulling together to work on the persistence of the SARS-CoV-2 virus in long COVID. She has been studying the persistence of measles virus, which I mentioned before, especially, the genetic RNA backbone of measles and she's doing a lot of this research in mice. She can actually show how that measles RNA is sticking around in the mice and even changing its activity over time to prevent the immune system from recognizing it, sometimes by inducing mutations in its own backbone so that it can better hide inside cells. And she's actually characterized these, unique mechanisms by which these viruses with the RNA backbones can persist. And so you can look at that work. There are coronaviruses, for example. There are studies. There was one team that found a different kind of coronavirus in MS brain lesion tissue before, if you look through the literature there. And then there have also been teams, for example, more recently that have connected viruses like dengue or Zika or Ebola to really clear chronic consequences. So, for example, post-Ebola syndrome is a condition that is similar to long COVID, where some people who survive Ebola go on to develop a range of chronic symptoms where they didn't die, but they're debilitatingly, chronically ill now. And what was key there in the study of post-Ebola syndrome that has a lot of relevance to SARS-CoV-2 persistence in long COVID is that teams looking at these Ebola survivors with chronic symptoms started to look in samples beyond their blood to see if they could still find the persistent RNA virus. And that's really key because part of the reason I think RNA viruses are understudied as chronic drivers of disease is because they're not often found in blood. It's just a dumb place to be. So if you persist in blood, that's where all the immune cells are, and that's where you're likely to get targeted and killed. So in simple terms, the RNA viruses will go into tissue and hide in tissue or in nerve in patients. And they're better protected from the immune response. And that's what we call a reservoir. So the post-Ebola community, almost right when COVID was beginning, was actually finding that some of these post-Ebola patients still had RNA in eye tissue, in semen samples, in breast milk, in samples where people had just not checked before to see if virus was even in those, because they were just basically looking in blood and being like, oh no, it's not there, it's not there. And so that led to an understanding that's growing now and an increased interest in studying RNA viruses, but we understand as drivers of persistence, but we understand that the types of studies and the infrastructure that we're building to study RNA viruses has to grow because we need to collect tissue samples from patients with long COVID, do more autopsy work where we examine tissue at death, or use a lot of forms of high resolution imaging where we can sometimes image viral products throughout the body and brains of patients to really understand what's happening in these patients. So there's history, but at the same time, and so there's precedence, obviously, and to summarize, there's precedence for RNA virus persistence from every virus, from dengue to measles to other coronaviruses to Ebola to even the enteroviruses, which I could mention more later, that are also, drivers of respiratory illnesses. But now with SARS-CoV-2 and long COVID becoming more widely studied, we're seeing an increase in interest in studying them all.
Yeah. I was wondering, could you maybe describe a bit on about your initial sort of reactions at the beginning of the pandemic and your concerns about these issues, and how quickly did it dawn on you that this question of long-term viral persistence and sort of long COVID would become a major societal problem?
Yeah, early. I think that I can't even remember ever thinking that long COVID wouldn't happen. It was just already an assumption in my brain. In fact, my husband and I were unlucky enough to get COVID as some of the first cases in Boston in 2020. And I remember trying to take a lot of supplements and resting and everything. I was already very, very worried that I could develop chronic symptoms from COVID. In other words, I was already thinking of trying to prevent long COVID, even in my own early 2020 case. So it was just a given to me because, again, every major pathogen really, once it's studied enough, has been connected to a chronic syndrome in which a subset of patients develop debilitating chronic symptoms. So it seemed very unlikely that SARS-CoV-2 would be an exception to that rule. And I think, though, that what was a defining moment for me in terms of thinking about SARS-CoV-2 persistence and really moving in that direction with our research program on long COVID was there's a team at Mount Sinai, Saurabh Mahandru's on the team who now we work with, and they published a paper in Nature where they had gotten intestinal tissue from patients after COVID. And these were patients just undergoing routine colonoscopy procedures, and they decided to intelligently look for the viral protein and RNA in some of the samples. And there's a beautiful image of RNA and protein in a sample from someone without symptoms, interestingly, an asymptomatic person, yet RNA and protein still there at 7 months. I think it's either 4 or 7 months now, too many studies blending in my head, but months after initial infection. And I thought, boom, there it is. Yeah. I'm not surprised because we know that the gut is a common reservoir site for other viruses, even HIV. And so from that point forward, seeing that image was a catalyzer to move in the direction of that research.
I know there's been just an immense amount of research done on both SARS-CoV-2, long COVID. There's now been literally tens of thousands of scientific papers published on these topics. And just on September 4th, members of the PolyBio Consortium published a very important position paper, which summarizes what we now know about the persistence of SARS-CoV-2 in the body after initial COVID-19 infection. You called special attention to the effects on the vagus nerve, the neurological system. I was wondering if maybe you could speak on some of the sort of key understandings that we now have about long COVID and viral persistence. Specifically, what body tissues have we found that SARS-CoV-2 can persist in?
Yeah, that's a great question. So as we delineate in the paper, the evidence for SARS-CoV-2 persistence thus far comes from a mix of autopsy studies where tissue is examined after death from patients who passed away. One particular study is an excellent example of that. I'll explain the study a bit more. A team at NIH, led by Dan Cherto's group, got tissue from patients who died had gotten COVID documented, but sometimes in one case, a person died 210 days after COVID from other causes. And they examined their tissue and looked throughout the many different types of tissue from the brain and the spinal cord and the guts and the lymph nodes, and did find viral RNA and in some cases protein throughout on a wide range of human brain and body sites. So that set a precedent because it's a beautifully done study too. The preservation, it's important when you're doing a study where you're looking for an RNA virus in tissue. You have to preserve the sample rapidly because otherwise the RNA can degrade and you can mistakenly say, oh, it's not there, but really the sample just degraded with the virus you're looking for. So that team did an amazing job just quickly freezing the samples and stabilizing. So it's a beautiful study. And so that sets precedent. And there's been other autopsy studies, for example, that have found SARS-CoV-2 in the lung or other sites after acute illness. In long COVID, obviously in living patients, you can biopsy only certain types of tissue. So there are teams, and that's part of what we're continuing to do now, who use a biopsy or sometimes a surgical procedure to get tissue from patients post-COVID. There's a team that found SARS-CoV-2 RNA in olfactory bulb tissue from people who had loss of smell after COVID, which is so basically connecting persistence of the virus in the olfactory bulb of the nose to loss of smell. Interestingly, those same patients test negative for the virus on the regular nasal swab test. They're negative, but the virus was found in the tissue of the bulb of the nose. And this is really key. The fluids of the body are not where you pick up the persistent virus. It's in the tissue. There's another interesting study that we were able to sneak into the paper at the last minute because it was published right when our paper was coming out, where it's another team at NIH. They got taste bud tissue via biopsy from the tongue of patients with loss of taste after COVID. And they found SARS-CoV-2 RNA, and in some cases protein, if I remember correctly, in the majority of the taste bud tissue of people who had loss of taste. And in a couple of cases, they track the people over time, and a couple people regain their taste a bit, and they did have clearance of virus. So it was really pretty correlated with the loss of taste being due to the virus still being in the taste bud tissue. Then there's a couple studies that have found SARS-CoV-2 RNA and protein in the gut of patients with long COVID symptoms or in stool, where long COVID patients are still studying the virus in stool samples, which is to me, not the best. Again, stool isn't really telling you what's in the tissue, so it's a proxy, but there's been some shedding in stool, even documented in long COVID. And then trying to think, one interesting study that we supported through our consortium did a cool analysis where they got gut biopsy tissue samples from 5 patients with long COVID, and they're actually continuing the study in more patients, which is great, and showed that all 5 of the patients had SARS-CoV-2 spike RNA in the gut tissue up to 676 days, I think, in one case, in one patient in the tissue, which was the last sample that's when they made the call to even analyze it, that they went ahead and did they could have waited longer. They might have found it for a longer period of time. And so those same patients underwent high resolution imaging that was able to document T cell activity throughout their bodies and brains. And a good number of them showed increased T cell activation via the imaging method in areas such as the spinal cord, the bone marrow, even the lymph nodes, which we think are an important site of reservoir on some of the tissue types that our current projects are working on, and the gut wall, which is not unexpected. So, and we don't know from those findings if the T cell activation is due to reservoir. It could be. It could also just be T cell activation, but it's interesting to start thinking about the possibility that reservoir could also be happening in long COVID patients in areas, particularly like bone marrow. That's a site we're really interested in now. We are beginning to work with a team at UCSF who's part of our consortium, and they're going to begin to collect bone marrow biopsies from long COVID patients. And it's going to be interesting to look at that site because the bone marrow is where a lot of immune cells are created. In other words, that's where platelets are derived from. So that's where neutrophils or other immune cells are first made before they're distributed through the rest of the body. So if there's persistent virus in the bone marrow, it can really become already integrated, including potentially spike into immune cells that traffic throughout the rest of the body. So it's an HIV reservoir site. Other viruses have been found there. It's no joke. We think it's important to study.
Yeah, absolutely. There's also could you speak a bit more on the findings on the vagus nerve and sort of the impacts on the nervous system as well?
Definitely. So I think the vagus nerve is a very important nerve that innervates every major trunk organ of the body. The word vagus is derived from vagabond, which means wanderer. And so the nerve literally wanders throughout the whole body, connecting to every major trunk organ. And then it enters the brain at the back of the brain in an area called the brain stem. And the brain stem is one of the most ancient conserved regions of the brain that regulates very important bodily functions, including autonomic signaling. So when you move from a sitting to a standing position, your body's able to regulate your blood pressure and make sure that you can do those postural adjustments. A lot of that autonomic signaling is controlled there. Also, an important response, so the sickness behavior response is controlled by the brainstem. And let me explain that a little bit more. When you're sick, let's say you have a food poisoning and there's a pathogen in your gut. Well, your body needs to know that you're sick. Your brain needs to know that you are sick in your gut, so it can tell you, in a sense, create flu-like symptoms and malaise type symptoms so that you rest or you lay down or don't eat more of the food, to alert you that there's sickness. So the vagus nerve is what drives that sickness behavior response. It can detect from gut or from lung and connect and drive these symptoms again, which are flu-like symptoms or malaise, that let people know they're sick. So we think then that if vagus nerve is directly infected or is picking up on SARS-CoV-2 reservoirs in the body and kind of conveying that inflammatory signal to the brainstem, that sickness behavior signal might never really turn off and might continue to signal you have flu-like symptoms, you have malaise, you have, which are prime long COVID symptoms. And if you combine that with the autonomic dysfunction and some other brain nuclei in that same region that control pain and nausea signaling, you get to a core of some of the main symptoms that long COVID people suffer from. So certainly either direct infection of vagus nerve as a reservoir, or the possibility that the vagus nerve is sensing reservoir in other sites and conveying that inflammation to the brainstem as a signal is a major area of investigation by our team, our groups, because the nerve is connected to so many classic long COVID symptoms.
Yeah. What other viruses have been shown to infect the vagus nerve or cause these impacts?
It's so understudied that in, I think it was 2011, my colleague at PolyBio, who's also a neuroscientist at Harvard, Mike Van Elzeker, wrote a paper hypothesizing that in patients with myalgic encephalomyelitis, ME/CFS, which is it's a related diagnosis where we knew before COVID that people who got other infections often develop common sets of symptoms, that it's another area, it's a chronic condition tied to infection. In those patients, Mike wrote this paper hypothesizing that maybe herpes viruses were infecting the vagus nerve in patients with those other viral onsets. And that started our thinking on vagus nerve, but very few people have actually looked for it because it's really hard to do. You cannot biopsy the vagus nerve. So we cannot get vagus nerve samples from any living patient. So it has to basically be done in an autopsy study or in an animal model, which is possible. One team did acute COVID autopsy study where they looked at vagus nerve, which is great. That's a great move to actually begin to collect the vagus nerve tissue. And they did find SARS-CoV-2 RNA in the vagus nerve of the majority of COVID patients in that study. So it's actually, Mike and I talk about this, it's so understudied as a nerve that could have persistent infection affect it, because what pathogen would not want, a lot of pathogens like infecting nerves. So why would they leave the biggest nerve in the body alone? The nerve that allows them to traffic through the body, from the brain to the body. It seems very unlikely that these pathogens, especially neurotrophic pathogens like SARS-CoV-2, would just not infect this kind of nerve superhighway that would allow them to reach more body sites. And in fact, one of my mentors, his name was Rob Moyer, and he studied viral seeding of amyloid plaque in Alzheimer's. He absolutely thought that the vagus nerve could serve as a highway for pathogens to traffic, for example, between the gut and the brain. And that's an interesting thought, because it would be entirely possible that someone, for example, could have SARS-CoV-2 persist in their gut for a while, but then slowly the virus creeps up the vagus nerve into the brain, and that's when you start to see some of the more severe cases. So it's definitely a nerve of interest.
Yeah, it's a very, very pernicious virus. I mean, just what you've presented is really, staggering. Just the widespread impacts that it can have on the body, and the persistence in all these different tissues. And my next question is sort of related to the cumulative societal impacts of long COVID. There's various estimates on the prevalence of long COVID, but in general, I think there's a consensus that somewhere from 10 to 30% of infections lead to prolonged symptoms. The risks of long COVID are slightly reduced by vaccination, but then they're also compounded with each reinfection. There were two recent surveys on long COVID, which gave potential estimates on the number of patients globally. The first, led by Eric Topol, gave a conservative estimate, which was based on official case figures, that at least 65 million people throughout the world are now suffering from long COVID. The second estimate was in a study led by Danny Altman, which noted, quote, the oncoming burden of long COVID faced by patients, healthcare providers, governments, and economies is so large as to be unfathomable. If 10% of acute infections lead to persistent symptoms, it could be predicted that about 400 million individuals globally are in need of support for long COVID, end quote. And I think, many long COVID researchers and advocates have really correctly referred to the pandemic as a mass disabling event. And I was wondering if you could comment on these figures and sort of the unprecedented scale of chronic disease that this pandemic has unleashed.
Yeah, I think that sometimes people will argue the numbers on long COVID. Is it 6% of the US population? Is it 7%? If it's 1%, if it's 0.5% of the population, this is still a monumental crisis. So part of me, I think that the point is first that even 1% of the population developing chronic symptoms after COVID is a huge, huge, huge problem. So, all the differences in those other stats are, less important to me. I think that it's important to consider that, I'm going to try to put the math together here. You have people who, this is the thing, is the definition of long COVID is very broad. So it currently encompasses people who develop syndromic type symptoms after COVID, but also people who have a cardiac event or a stroke or a pulmonary embolism. And so there's a lot of different events that are being pulled into that long COVID number. They all matter, but the cases of people who are developing a new onset disease are probably a bit of a smaller subset of those wider statistics. And yet all of it matters, because even beyond the numbers of what's being documented as long COVID, I don't know about you, but when I talk to friends, for example, I have a couple of friends where one of my best friends has almost a photographic memory. In high school, we would get in trouble and then she would help find our way home when we were driving back, and she would always have every street memorized, and it was amazing. We called her the Brain. Literally, that was her nickname. And she told me that after COVID, she just can't remember things like that anymore. She just kind of lost that ability. And she doesn't consider herself to have long COVID. So she's not included in that diagnostic label, but she did have chronic effects from it. And I think that's what is also really concerning, is this isn't just some people getting long COVID and the chronic effects and everyone else doing perfectly fine. If you look at the research, you'll actually see in the studies that a good number of times the control subjects are the healthy people. So in every study, we take the long COVID patients and we compare them to people who are healthy. Sometimes those are people who got COVID and recovered and don't report symptoms. We call them convalescent controls, and they returned to the convalescent phase. Sometimes those people who don't report symptoms still have spike protein in their blood or immune activation. And what's concerning about that then is that it might actually be possible that even people who don't manifest symptoms right away after COVID still have some biological abnormalities. For example, maybe didn't fully clear the virus, but just don't have symptoms yet from that. When herpes viruses persist, they often persist dormantly without symptom manifestation for a while. So the concerning part is that wide scope of continuum between people. I mean, the extra concerning part is beyond the specific long COVID numbers. You're getting an increasing number of people who just might have biological problems even without symptoms. That T cell activation study that I mentioned before, some of the controls are the people who didn't report symptoms also had T cell activation in that study. In fact, the real comparison in that study where you saw the most T cell activation versus not was from people before the pandemic and after the pandemic. So overall, we're looking at a situation in which a good number of people are developing symptoms or signs of biological problems after COVID. And people are getting sick sometimes 3 to 4 times a year, and they're getting infected with different variants, which are wonky and immune evading. I mean, I think it's interesting when you hear, oh, look at the new immune evading variant. Well, you know what facilitates persistence of a virus? Evasion of the immune system. So it's not, we barely even are able to study all these variants. They emerge so quickly, but the fact that they're evolving around the human immune response is not necessarily a positive sign for their clearance. And then you have children getting sick. So there's a study done by a group at NIH where they got tonsil tissue from children who are undergoing routine surgery. They were just getting their tonsils removed. Now, you have to question why they were getting their tonsils removed. There must have been a reason. So sometimes tonsils are removed because of breathing problems. So maybe some of the kids did struggle with some kind of post-COVID breathing problem, but that wasn't documented as part of the study. They just got the tissue from the kids undergoing the surgery. And when they look at this tonsil tissue, which is a type of lymph node tissue, they find viral SARS-CoV-2 RNA in a good number of the samples from these non-symptomatic children undergoing routine surgery. And their immune responses, the B cell responses that these children had, were abrogated as well. So the immune response had been thrown off somewhat because it's still responding to that persistent RNA. So we're seeding children from a young age with a viral RNA in their tissues, with a virus that people are getting multiple times a year as it continues to mutate. And we do, as you mentioned, see that reinfection seems to up the chance of developing chronic symptoms. It's just absolutely unsustainable and a complete crisis.
Yeah, absolutely. I think those are very powerful remarks. And I was going to ask actually about this. There's the essay that was published by the John Snow Project, which raises many of these questions. And I think that just the work of that project has been very important. Really, it's an excellent source of content on the pandemic and long COVID. The videos and the papers that you all have published have been really fantastic. But this September 14 paper, COVID-19, a new disease paradigm, pointed to this study, which was led by Michael Peluso on the persistent T lymphocyte activation, and also these what you're raising about asymptomatic acute infection and asymptomatic viral persistence. The paper notes sharply, quote, the issue we face is we may not know the full extent of harm until it is too late to remedy it. And in the context of a virus that has infected most of the global population, including children, continues to circulate and reinfect, this would seem to be an unacceptable risk, end quote. So yeah, I guess I was wondering if you could speak a bit more on the John Snow Project, and also maybe elaborate a bit more on some of your, like, what are your greatest concerns about long COVID and viral persistence?
Yeah, the John Snow Project is a group of scientists and some members of the community who think that COVID still matters, but are interested in finding solutions, new solutions together as a team, and also in helping the public understand some of the nuances of COVID that have not really made it through in some of the regular news stories that you hear. With one of the ones, and this is part of why I joined the group, one of them just being how the virus is circulates in the air. I think one of the most undoubtedly criminal aspects of this pandemic has been that our public health authorities have not told people how the virus spreads. They, from the beginning, we remember the 6 feet rule. I still see signs in stores that have a thing of reminding people to stand 6 feet apart, or people wearing a surgical mask. Yeah, where, the mask has gaps where air can come in.
Baggy blues.
Yeah, exactly. That is a tragedy, because the virus is in the air. It's an aerosolized virus, and anyone who's actually studying the virus well can tell you this. And what that means, and John Snow team did a really good video on this, is it spreads through the air like cigarette smoke. So people are in a room, it's just beginning to fill the air in the room with, and if you don't open windows, and if you don't do anything to mitigate that you can just get it, even though you're, 6 feet from someone, right? It's so few people know that. It's just incredibly painful. There's a nursing home right by where I live, and I go on walks in the morning, and there's a sign at the nursing home that says, wash your hands when you come in, please, for your COVID measures. And we know COVID doesn't spread much on surfaces. I don't think that they actually know that people can come in, wearing a surgical mask and spread that, and that's a tragedy. So the John Snow Project is working on helping people understand that the virus spreads in the air, but also working on potential solutions to clean the air. And we do think that's important because as long COVID researchers, we are going to burn out. I want people to know this. We can study the chronic patients, and we can try to open more and more and more and more and more clinics. And we can try to run clinical trials, which we are, of combination therapies to see if we can get the virus out of tissue from a nerve where we're going to need almost certainly multiple drugs and long courses. And we can do all that. But if we do not do that in concert with basic efforts like infrastructure to clean the air so that people are less likely to inhale this virus in the first place, I don't actually see a solution. So part of our consortium this year, part of our goal is teaming up with scientists who are creating affordable air quality solutions, HEPA filters, germicidal UV technologies that can clean the air, and involving them in our discussions and our meetings, because we need a multi-pronged approach to be able to deal with this virus now. And that's part of what the John Snow Project gets, which is really cool to be part of.
Yeah, no, absolutely. I think those are important points, and I really commend the work that that you all have done. It's been very significant, and I think we have to really sort of expand this advocacy throughout the pandemic. The WSWS has also advocated for a strategy of global elimination, and really, the use of all public health measures to really contain the virus. And I think these points that you're making on, cleaning indoor air are critical, and it really, requires massive societal resources, the investment in infrastructure, in HEPA filters. I think doing more clinical trials on, UV lamps and so on, and really encouraging masking.
Yes.
And yeah, and I guess I did have a question. I think I was wondering if you could comment on sort of the role of the corporate media during the pandemic, and sort of specifically, the lack of coverage on these issues, but also long COVID. There's really for over 3 years now, researchers and patient advocates have sort of been ringing alarm bells about long COVID, but it's basically gone unreported in the mainstream media. NBC, CBS, ABC, CNN, they all have vast resources, but they've never, allocated an hour to really doing a deep dive into the science of long COVID or airborne transmission. And maybe if you could share your thoughts on this issue.
It is frustrating, and I don't watch mainstream media, so I feel like I actually almost started to follow some of the mainstream outlets on purpose to try to understand. I actually do this now for a number of topics, trying to figure out what the outlets are saying so I better understand what some of my friends and family are hearing, so that I know sometimes when I can come in and actually need to inform them that something, there's additional information that they might want to know. I think that with the pandemic, the whole pandemic has been a problem with mainstream media. So if everyone's over COVID, they're certainly over long COVID. This is the problem that we have. I mean, I understand. I understand. I think that we lost some faith in public health with this pandemic because it wasn't very well handled. Early on, for example, people were not given a clear message to wear N95 masks. And it's really confusing for people when they realize that they weren't given the right information. We also went with a vaccine-only approach, which was oversold. I mean, vaccines can be part of the picture, but we needed to explain more to people that masks and what, exposure and just even other kinds of spending more time outdoors instead of inside, opening your windows. We failed to tell people a lot of basic things that could have been important as part of the solution, and threw a lot onto the vaccines. I think that we weren't in a great place with public health to begin anyway. We're living in an epidemic. This is what's the worst part. We're already living in an epidemic of chronic disease. Before even COVID began, look at the skyrocketing rates of conditions. Look at Alzheimer's disease. I mean, rates of Alzheimer's, I'm pretty sure the statistic is that someone gets Alzheimer's every 65 seconds. I mean, people have gotten, 20 people got diagnosed since we've been talking. It's completely out of control. And the investment in the Alzheimer's space has all gone in the wrong direction, with drugs trying to remove the amyloid plaque from the brain, when actually the plaque is now being studied as part of the immune response to pathogens that can persist in brain tissue and potentially seed the plaque. So the plaque has a purpose. That's what we're figuring out. And we've been trying to remove this plaque the whole time. But you'd understand this, once pharma goes down this path of they started to develop amyloid removing drugs, and every company that had theirs in the pipeline had to get back to their investors, had to get their return on investment. We start these cascades of chronic disease problems that are not solved because of, in some ways, profit motive and things like that. So when COVID began, we already had a burdened population. And I think that we had to message people in a way about COVID that was more realistic. I think we needed people to talk about COVID who would just actually talk to people more directly as intelligent people capable of understanding nuance. There's messaging in public health, which can be like, people are, kind of sheep. We can only tell them simple things. No, we should have sat down, and there should have exactly been a frank discussion of the risks of getting COVID, what it entails, how to make a decision. Because people did have to work. In other words, ways to help people sort through these questions. I do understand. I have friends whose businesses shut down and they've never recovered since COVID. I know people who had children who they didn't go to school for over a year, so they still have trouble with friends. I mean, there are social consequences from shutting down, and we need to have an honest conversation about that, but also about the risks of the virus, including the chronic consequences of the virus. And I feel a kind of honest discussion would have led more to people being willing to think about it more. But there's too many sound bites and too many overconfident assumptions, in a way, half of which sort of proved wrong, predictions that didn't work out, and people burned out on it. And in a way, I understand that's the worst part. And so now when people are burned out on COVID, the last thing they want to hear about is long COVID. I mean, although they should, if anything, when talking to a younger demographic of people about why they would care about COVID, it is really important. And it does make people think more when you say, look, it's not just death. It's not just if you live or die. The truth is, the bigger problem, especially for people who are younger, is just whether you're going to get your life back, if you're going to, not have chronic disability from this, to the point of, and the effect now on the workforce. For example, there's a serious drop in workforce over this, people who are sick, and people don't know that until it's too late. And so, yeah, I don't know what the remedy, thinking for the remedy of this. We have to start to bring COVID back up again. And maybe the data that we're creating will help. That's part of why we're doing what we're doing. The reason we're running the long COVID consortium that we're running, which is a group really of top researchers in the world working together, and actually working in close collaboration. So sharing ideas and samples and legitimately and working openly. And one of our donors is Vitalik Buterin, who is a champion of open source, open science and open source drug development, which is critically important. There is no way that we are going to come up with solutions for COVID if everyone's just sitting on their ideas in separate labs, waiting to see if they can patent them. There's just no way that we will work in the face of this emergency unless people are actually openly sharing ideas and resources. And that's what our consortium does. And the goal is that we will and are beginning to produce these heavy hitting papers, really showing like, there's the virus in tissue, there's the virus in the gut, there's the virus in your brain, there's the virus, to be able to hold up a growing body of evidence to people to be able to see the damage that's happening. That's why we're doing what we're doing, so that hopefully maybe it can be brought to the attention of the public in a transparent way to begin to up the narrative again of what we're supposed to do about this COVID situation.
Yeah, no, I think those are very important points. And I think the international collaboration of scientists is also critical. And really what we fought for is really to try to bring this into the working class, to educate workers. And anytime we host an event, we encourage masking. We use HEPA filters, and we've always found that there's a very strong response. Yeah, just earlier this week, we had a meeting at the University of Michigan where we asked audience members to wear masks. We provided N95 masks, and essentially 99% of the audience did. There's no opposition. And I think when it's explained and the dangers of long COVID are explained, people are more than willing to both protect themselves and protect others. But really, we have to, there has to be a major increase in the spread of this, scientific information and, the dissemination of this knowledge into the working class. And just, I guess my final, or two more questions. Just one is, it was just over a year ago that US President Joe Biden declared the pandemic is over, the auto show here in Detroit. And then this really sort of, this was part of a deepening propaganda campaign, and it sort of culminated in the ending of the public health emergency declarations by the WHO and the Biden administration in May, which sort of prompted the scrapping of all data collection and tracking in the US and internationally. And we saw how premature this was, and the fact that just 2 months later in July, we saw yet another summer surge of infections, which you can now only see through wastewater. And we're seeing, as you said, these new variants continue to evolve, the danger of Proal and other variants. So I was wondering, could you comment on these issues and sort of the dangers of declaring the pandemic over while the threat of long COVID, remains high for everyone?
It's terrible. And to be honest, incredibly hypocritical, because I'm not sure, but there's videos out there, when Biden goes and gives a talk somewhere and he has no mask on and he's speaking to an audience, there's a big setup behind him that you don't see in the video, where there's a bunch of HEPA filters and all kinds of things actually purifying his air that they don't even let the viewer know is happening. And that is incredibly hypocritical. So in other words, they do recognize and understand that SARS-CoV-2 could be right there. They're actually just not communicating that fact. That's brutal. So yeah. No, there's no way. The pandemic is not over, and it's really strange when people, will say during COVID, this is a constant thing where friends of mine, and it's hard when you're studying long COVID, and a friend will say, it was hard during COVID when this, and I'll have to say, it's still COVID. And I don't want to have to do that anymore. We need leadership that is, again, transparent and realistic about COVID. I don't think that, we need a conversation with people about how this should be handled. We need to think it through, but we need someone as a leader who does understand that a virus that continually circles throughout the population and is capable of driving so many chronic consequences is something that must be explained and hopefully dealt with somewhat.
Right. I would just say, I don't think that leader is going to come from either the Democrats or the Republicans, but
no.
I guess my last question is, I was wondering if you could discuss just some of the treatments that are being investigated by researchers with PolyBio or other research groups that you're aware of? And I know it's hard to predict, but what do you think is sort of a realistic timeline for developing treatments for long COVID?
Well, with viral persistence and reservoir of the virus in tissue, there are 4 Paxlovid trials underway. Now, whether Paxlovid will work to clear persistent virus is an open question. The mechanism of Paxlovid, that we're not sure. The tissue penetration might not be good enough to get to reservoir. Also, the length of dosing. The trials are somewhat short in the scope of trials used to treat persistent infection. One, I think, is 15 days. That might be enough, but actually longer courses of any antiviral might be needed in the treatment of reservoir. Everyone kind of knows, though, that those monotherapies or single, whether it's an antiviral or even a monoclonal antibody or an immunotherapy, might work on their own. But like, for example, with HIV, where it's also treatment is used to contain a virus, multiple drugs are needed. So combination clinical trials are going to be really important. And certainly there's a group, a trial that we're partially supporting at UCSF of a monoclonal antibody, where they got a company to donate a monoclonal created during the Delta wave of COVID, and it's patients who got long COVID during that same wave. So hopefully the monoclonal is matched with the virus that might be persistent in those particular patients, as kind of a proof of concept that we could bring in monoclonals to basically bring the immune system back up to potentially get persistent virus while also wearing it down with antivirals or other drugs. So there's movement in the space, but right now NIH doesn't have much of a infrastructure to fund clinical trials. So you'll see a couple come out of, again, our privately funded group, but we need much more large scale government commitment to the actual infrastructure.
Yeah, no, absolutely. And I think it sort of really goes to the the need for what we advocate for is really a socialist public health program, and really massive investment in public health. Or Biden is demanding over $100 billion for the military and for war.
Exactly.
But there's nothing, just a pittance for public health and for long COVID research.
It's incredible. I forget, but I think we make something like 12 nuclear submarines every year, each at the cost of trillions of dollars. Well, let's make 10 of them instead of 12 and take those trillions and put it into long COVID. I mean, this is not hard. Just pull a fraction away from the military industrial complex and we're good to go. It's because when people say, where would the money come from? Yeah, how about that? Let's actually work on our infrastructure here, cleaning the air, building technologies to help study long COVID. We could invest here instead of with picking wars in other countries. So I agree with that.
All right. Well, thank you so much, Dr. Proal, for doing this interview today, and for really the tremendous work that you've done throughout the pandemic on the work of PolyBio.
Thank you very much.
Thank you.
All right. Bye.